Welcome to another lecture on quality control. Today we'll be looking at principle of quality control of culture media. Now, ensuring accuracy and reliability of scientific data relies on meticulous maintenance of pure, consistent, growth supporting medium through rigorous quality control principles to provide the desired results. So, in principle of quality control of culture media, there are certain guidelines, certain procedures that must be followed in order for us to get our desired microbial culture. So we'll be looking at this in a brief. Microbial culture media quality control is very important for the accuracy and reliability of experiments and diagnostic tests. It is very crucial that this is carried out because the principle dictates a certain procedure to cultivate specific microorganisms under optimal conditions while mitigating contamination and generation of erroneous results. So, we will look at the major aspect of quality control of media under the following headings: sterility, composition and formulation, physical characteristics, performance testing, and documentation and traceability. Sterility. To ensure accurate results, culture media demands complete sterility, void of any contaminants, because we are specifically looking at a target organism. Hence, the media must be sterile to ensure that any organism that grows is what we are specifically looking at. And this sterility is achieved through methods such as autoclaving, mostly, or by any other means of sterilizing the media in the laboratory to eliminate contaminating microbes or their spores, which can overgrow or shield or mask the target organism, because your target organism may be a slow growing organism. Hence, elimination of potential contaminants which may grow faster than the target organism must be ensured, and this, if not correctly carried out, may lead to false identification or misleading data in the process of experiment. Then the composition and formulation. Every microbiological culture media are tailored constituents or ingredients that has nutritive value in nature and solidifying agents plus selective elements which are designed to give the microbial cells a feel of their natural environment to mimic their growth requirements for specific microorganisms during isolation, identification, and study. So, this composition and formulation is essential for guaranteed success and identification of result every time. Precise weighing and measurement of ingredients following the standardized constituents to ingredients quantity must be ensured to provide consistent growth of the target organism. If the constituent or composition of the formulation is below what is expected, this will be a requirement for another organism which can be in the environment. Hence, there will be a false growth of organism. There will be a false result leading to unwanted organism or certain organism that are versatile in the environment or probably as a means of contamination, which may now grow, and this will lead to a false result. And every microbiologist's desire to grow the target organism in every experiment and avoid contamination. So the formulation is essential. The composition and formulation must be adhered to strictly. Then the physical characteristics of the agar, which includes the pH, acidity and alkaline nature of the agar, the concentration of the agar, which can determine the thickness of the agar, the color content, which can be a form of, the color indicator, which gives the agar medium particular reaction based on microorganism fermentation or pigment production, has direct influence in differentiating several microorganisms during cultivation. When we deviate from the expected pH by altering the pH content, we give room for variation in the physical characteristics of the agar, and this can lead to false results. Hence, physical characteristics of every agar must be controlled to give a quality growth of the target bacteria. In certain instances, visual inspection and measurement tools are used to ensure that the media meet the required physical specification before this media is now deployed for use, or before it can be sold, or before it is being, autoclaved for cultivation of microorganisms. And the physical characteristic is very important. Then, the performance testing. Evaluation of media suitability for target organism cultivation is achieved through growth promotion testing. These assessments involve inoculation of established reference strain. We have different reference strains, we have ATCC, we have MTCC, and so on, onto the test media, followed by strict comparison of their growth pattern against well-defined benchmark of microbial growth of the same species. So the efficacy of selective media is rigorously assessed through selectivity and differentiation tests, which ensures that their capacity to suppress contaminants, that is unwanted organisms, while promoting the growth and expression of specific characteristic feature in the target organism is ensured. For growth promotion tests, it is a quality control requirement that confirms the ability of a new batch of media to support growth of a predetermined selection of representative microorganisms in every setting. In terms of quality control of microbiological media, we can discuss it in 4 headings: control of preparation, testing of physical and chemical parameters, group promotion testing, and control of storage condition. We will look at this in different form. We look at this one after the other. Now, control of media preparation. For every culture media that is used in the laboratory, it can be acquired in 2 ways. First, it can be acquired from an established manufacturer. So when we want to control media preparation, if the media is obtained from a well-established manufacturer, such as Oxoid, such as Biolab, and so on and so forth, high media, we are purchasing and relying on the integrity of the manufacturer. This requires a trust from the manufacturer. So whatever we are preparing, we believe that every quality control assurance has been met on the integrity of the manufacturer. If we don't purchase, if we want to prepare our own media, which is a compounded media, also known as in-house manufacture with specific ingredients, then our in-house media relies on certain specific measurements in terms of raw material that is going to be used, must be adequate, must be specific, and must meet up the standard or must be suitable for the microorganism that we are targeting. Remember, when a media is being prepared, there's a microorganism that is being prepared for. Then storage condition. The raw materials, including the media that's being prepared, must meet up with the standard storage condition so that the media may not lose its texture or may be dried, over dried, or may be destroyed by improper storage condition. Then preservation of the material for the media is also very important because every material for the media, such as ingredients, nutrients, elements, and so on, must be properly preserved to maintain their nature. Otherwise, there will be a preparation of a media which has deficiency in one or two factors that is expected to be present. So the goal of this testing is to provide a gatekeeping function before investing time in growth promoting testing. If any of this is not ensured and the growth promoting testing is carried out, the result is bound to be failed. To be a failed result because the growth promoting testing relies on the integrity of the media. Integrity of the media relies on the integrity and quality control of whoever is preparing this media. So after this is done, then we go to what is called the growth promoting testing. The growth promoting testing consists of 3 methods. These are methods that are used in determining the growth of microorganisms in a particular medium. It is a microbiological quality control procedure. It is used in verifying that a new batch of agar medium can adequately support the growth of the target organism, which is intended for, and it will ensure that there is accurate result in subsequent microbiological tests. Now we have 3 methods that are being used popularly. We have the pop method, spread plate method, and the spiral plate method. I will explain this method one after the other. The first method is the pop plate method. The pop plate method is a technique that is used for the enumeration and counting of a viable bacteria in a medium. Most of the requirements: petri dish, the broth containing microorganism, the agar, the Bunsen burner, and so on and so forth. Now, every microbiologist knows that before you carry out any culture, you have to sterilize your workbench and sterilize your hand, put on your hand gloves, just like the person in this picture. Then the preparation is done by carrying out a serial dilution of your target microorganism after incubation. After standardizing culture to something like a McFarland standard, or depending on the number of serial dilution diluting factor that must be used. Then we proceed to prepare 0.1 ml of the culture onto an empty sterile petri dish. Then a molten agar, about 20 ml, which is usually the highest for the standard petri dish, is added onto the dish containing this sample, but it must be allowed to be cooled to about 45 to 50 degrees centigrade before adding, so as to avoid killing the bacteria. Then this molten agar and the culture is gently swerved to mix the sample and agar together without creating bubbles. You must avoid bubbles while mixing this agar together, because you have to do it gently. If there's any case of bubbles, once it's solidified, it can alter the view of the agar or even the result. So the agar is now allowed to solidify after 20 minutes. Then ensure there is no bubbles. The agar is now inverted and incubated in the incubator for the specific required temperature and growth condition for the organism. Different microorganisms have different growth conditions, such as temperature, oxygen content, and so on. So when the organism, after 24 hours to 48 hours, depending on the type of organism and the growth condition, the colonies can be seen, visible, and will be counted on the surface of the agar. So based on the reference strain on this particular agar, there's a comparison to show that this agar has actually passed the growth promoting test, certifying that the agar is good, can support the growth of the specific target organism. That is for the pour plate method. The pour plate method can also be carried out by inoculating the same 0.1 ml inside McCartney bottles that is containing the molten agar and poured together. It can also be done that way. Then we look at the next method. So for the spread plate method, we go on with the same procedure, except that in this case the agar is poured, the molten agar is poured on the petri dish and allowed to solidify. After solidifying, then the same quantity of the culture, depending on the procedure, sometimes we can have 1 ml or 0.1 ml added on the surface of the agar. Then with the use of a glass spreader, the culture is now spread. Remember, the organism has been diluted by your serial dilution. So the final dilution is now taken and added to the surface of the agar and spread with a glass spreader. This method is usually used for fungi, call it fungal carpeted growths, and also used for bacteria, but it's most good for fungi, and incubated. The same procedure for incubation, then enumeration. The third method is called the spiral plate method. It's a wonderful technique that is used in microbiology to enumerate or count the number of viable bacteria, just like we did for the two methods. So count from a liquid sample, usually from the broth. It has other advantages over pour plates and spread plate, because it is specialized in dispensing a known volume of sample onto a rotating agar plate in a spiral pattern. After the incubation, bacteria grow at different density around the spiral tract, and by counting the colony within specific area of the plate with appropriate dilution factor, the total number of bacteria in the original sample can be calculated. The advantage this method has is that it's faster, it's more efficient, it requires less sample and media compared to other methods, saving resources and costs. It's faster because it provides results quicker than other methods due to built-in dilution. It has a built-in dilution and reduced need for multiple plates. Then it also has a wider coverage for bacterial concentration on a single plate. So it reduces the need for multiple dilutions, and it offers a highly standardized and reproducible technique during the assay. So this is how it will look like after the results. It will look like this, although this may not be exactly the way it is, but the growth pattern will form in a spiral form. It is a good method, and like I said, it offers a faster approach to other methods. Then for group promoting testing of broth media, it involves a crucial quality control in microbiology that ensures the suitability of broth for supporting microbial growth. It provides sterility testing, microbial enumeration, and isolation identification of specific microorganisms. So the broth is what is used for the testing on pour plate, testing on spiral plate, and so on. So this broth must be prepared, and the broth media also ensures that the microorganism is supported and can be grown. So this is a picture of how a broth looks like in a test tube. Then group promoting testing of broth media itself has different approach by employing copious growth culture to rigorously assess the inter-batch consistency of media preparation by emphasizing on the reliability and control measures, while endpoint methods offer a straightforward approach to evaluate broad media growth promotion capabilities by comparing them to established standards by using standard strains. So their implementation presents significant technical challenges and requires substantial manual effort. The MPN, which is the most probable number enumeration of microorganisms, find particular utility in QC laboratory, quality control laboratory, when sample characteristics such as clumping or complex matrix preclude its conversion into homogeneous suspension, which are amenable to direct counting or filtration methods. In this type of study, in this type of experiment, it aims to investigate the critical dilution threshold beyond which the unknown population density renders successful inoculation of growth media effectively limiting the sensitivity of the most probable number MPN technique. While the kinetic parameters growth curve allows us to determine how the organism grows per time, and this is measured through the use of spectrophotometer that allows us to determine the turbidity and interpret it on a growth curve that is plotted on a graph. So the kinetics parameter is started at 0 time. First of all, at 0 time, at 0t, that is this time of inoculation. After 1 hour, then we check 60 minutes, then 120 minutes, then 180, and so on. So from 0 time to the 1 hour, 2 hours, 3 hours. So this growth is now measured, how the organism is growing, how supportive is the broth to allow the organism to multiply. The culture are enumerated and plotted. So it is expected to have a curve, growth curve, that grows gradually and increasing in height continuously, at least for the number of hours that's expected to be checked. And this will allow to determine the viability, the supporting ingredient present in the broth during this time. So for control of storage, the lab needs a system to keep untested media separate from tested media to avoid contamination. Ideally, a dedicated storage room should exist with a clear marking and storing of quarantine media within the same room. This is an acceptable pattern in every microbiology lab. There must be a room where quarantine media is kept. So all media must pass all quality check before use, and they must be stored under the same condition as approved by the label or by the protocol or SOP for each laboratory. Then we control the storage of condition. Media storage requires control condition, which is temperature, light, humidity, and so on, to maintain quality until the expiring date of the media is achieved. So until this, the media must be controlled in a conducive storage condition to avoid decaying or caking of agar. While not all factors apply to all medias, not all factors like temperature, light, humidity, they are not applicable to all media. Different type of media have specific requirements. For example, like the TSA, 50K soil agar, is tolerant of various conditions, unlike some other agar, which are effervescent, can easily absorb water from the environment or can easily solidify at higher temperature. So a careful look, just the way the scientist in this slide is giving, a careful look must be ensured to see if the media is still in good condition. However, in such careful look is not when you are holding the petri dish, it is before the media is prepared. So Thank you for listening, and have a wonderful day. Till we meet again. Thank you.